When you think about the world's most terrifying diseases, your mind probably goes straight to the movies.
Oh, absolutely.
Like you picture the hazmat suits, the sooty bleeding from the eyes, just this total biological meltdown happening in like a matter of hours.
Yeah, it's the ultimate villain narrative, really.
Right. Because it's binary. You're either totally fine or you're this Hollywood monster.
Well, we like our threats to be highly visible. We want them to fit neatly into a three-act script where the danger is just obvious from a mile away.
Exactly. But then, you look at the actual clinical reality of the Ebola virus, and suddenly that whole movie script just gets completely thrown out the window.
It really does.
We are looking at a viral landscape that is far more complex and honestly much more terrifying than fiction. So, welcome to today's deep dive. We are taking you on a journey to strip away those Hollywood myths, unpack the gritty reality of how this virus actually operates inside the human body, and explore the incredibly complicated scientific breakthroughs that are finally giving us a fighting chance.
Yeah, and we're grounding this whole conversation in two massive recent reviews.
Right, one from the Lancet and another from the New England Journal of Medicine. These are, they're pretty much the gold standard for where our understanding of the virus sits right now.
They really are the gold standard. And what those sources make immediately clear is that to truly understand this virus, we have to look really closely at the 2013 to 2016 West African outbreak.
Yeah.
That was the pivotal event that kind of shattered a lot of our basic assumptions.
Because before that, Ebola outbreaks were generally thought of as these really isolated events, right? Like in highly remote rural villages.
Exactly. But West Africa showed us an unprecedented scale. I mean, over 28,000 confirmed cases, more than 11,000 deaths. It jumped into major urban centers, it crossed borders, and it just crippled regional economies.
Okay, let's unpack this because
Yeah.
if we're going to fight a monster capable of that kind of devastation, we have to understand exactly what it is.
That's right.
And, I was reading through the section on the virus's family tree, and the taxonomy alone is kind of a maze.
Oh, it is definitely a bit of a maze. So the Ebola virus belongs to a larger family called the filoviridae family. They are filoviruses.
And philo refers to their shape,
Yeah,
right?
exactly. They are filamentous.
Right.
If you look at them under an electron microscope, they don't look like those round, spiky spheres we see with a lot of other viruses.
Like the classic virus picture.
Right, exactly. Instead, they look like long, tangled pieces of thread, sometimes looped at the end like a shepherd's crook. And they are non-segmented negative sense RNA viruses.
Which means their genetic code is written in a single strand of RNA. But it's basically in reverse, right? Like the host cell has to translate it before the virus can even start copying itself.
That's the mechanism, yes. It carries its own enzyme to transcribe that negative strand into a readable positive strand once it, gets inside our cells.
Got it.
Now, as for the name itself, Ebola virus, when it's all one word and capitalized, that is the overarching genus. But when we say Ebola virus with a space between the words, we are talking specifically about the virus that causes Zaire Ebola virus disease.
Okay, so within that single word genus, there's a whole family tree of different species.
Right. There are 5 distinct species that we've known about for a while: Zaire, Sudan, Bundibugyo, Thai forest, and Reston.
Okay.
And Zaire, Sudan, and Bundibugyo are the ones responsible for the major human outbreaks.
Right, but here's where it gets really interesting for me, though. I was looking at the Reston strain, and it completely threw me off.
Oh yeah, Reston is fascinating.
Right. The sources note it showed up in monkeys imported into the United States from the Philippines back in 1989. And then later it was found in pigs in the Philippines, and sequences were even detected in pigs in China.
Yep.
How does a filovirus jump across the globe, end up in the American suburbs and Chinese farms, and not cause a global medical meltdown?
Because Reston is this bizarre biological anomaly. I mean, it circulates in animals, it spreads. People handling those animals absolutely get infected.
Wait, they do get infected? Meaning we get the virus in our system, our bodies react, but we don't actually get sick.
Exactly. We don't develop the disease, our immune system handles it, or the virus simply, lacks the specific molecular keys to cause massive damage in human cells, even though it's highly lethal to monkeys.
Wow.
And the filovirus family tree is still expanding, by the way. We've recently sequenced genomes of entirely new filoviruses in bats, like the Loviu virus in Europe and the Bombali virus in Sierra Leone. And incredibly, researchers have even found filoviruses in fish, Stryavirus and Thamnovirus.
Fish? Seriously? So this thread-like viral architecture is much older and way more widespread in nature than just a few isolated jungles in Africa.
Absolutely. It points to a deep, deep evolutionary history. And every time we find a new one, it raises a really important question about zoonotic potential.
Meaning the ability to jump from an animal reservoir into humans.
Exactly.
Let's talk about that jump, actually,
Yeah.
because the sources dig into this idea that fruit bats are the primary reservoir for Ebola. But reading closely, it seems like we have never actually caught a live bat in the wild, tested it, and found the complete live Ebola virus actively replicating in its system.
That is correct, yes.
So wait, if we've never actually caught a bat red-handed with the virus in nature, are we just guessing? I mean, it sounds like trying to convict a suspect when the murder weapon is completely missing.
It's a frustrating reality of virology. We have very strong circumstantial evidence, right?
Yeah.
We find antibodies against Ebola in bats, and we find fragments of the viral genetic sequence in bat tissue.
But not the live virus itself.
Right. Isolating the live virus from a wild bat is incredibly difficult.
Why is that so hard, though? Like, if they are carrying it, shouldn't it be obvious?
Not necessarily. First off, the viral loads in the bats might be extremely low. They aren't getting sick, so the virus is just quietly persisting at barely detectable levels.
Oh, I see.
Second, to grow a virus in a lab, we have to put it into cultivated cell lines. And the cell lines we traditionally use might not be the right environment for a virus that's adapted specifically to bat biology.
That makes sense.
And third, bat tissue might actually contain natural inhibitors that neutralize a virus the moment we try to extract it. So, bats remain the primary suspect, but the actual spillover event, where a human handles an infected bat, or maybe comes into contact with fruit contaminated by bat saliva, it's a very rare, highly complex intersection of biology and ecology.
Okay, so the stars align in the worst possible way, and that rare jump happens. The virus gets into a human bloodstream. Let's trace what happens next, because this is where the movie version just completely falls apart.
Oh, totally.
How does a little tangled thread of RNA take down a human?
The physical break-in is honestly fascinating. So the virus is covered in a viral spike formed by a specific glycoprotein. And when it bumps into a host cell, usually an immune cell like a macrophage first, it doesn't just punch a hole.
Okay.
It tricks the cell into swallowing it. The process is called macropinocytosis.
I've heard that translated as cell drinking, right? So the cell thinks it's just taking in surrounding fluids and nutrients, but it accidentally gulps down a Trojan horse.
That's exactly how it works. The virus gets engulfed in a little bubble called an endosome. Now it's inside the cell, but it's strapped in this bubble. Here the environment is acidic, and host enzymes start chewing on the viral glycoprotein.
Trying to digest it.
Right, but this cleavage actually exposes a new part of the virus, which binds to a specific receptor on the inside of that bubble called Niemann-Pick C1.
So it uses the host cell's own digestion process to literally pick the lock.
Yes, exactly. Once it binds to Niemann-Pick C1, the viral envelope fuses with the bubble, and it dumps its RNA directly into the cell's main compartment, the cytosol. Now it has control. It just starts aggressively copying itself.
And as it's doing this, the host cell has no idea. I mean, doesn't the cell have alarms it can pull when it senses foreign RNA?
It does. Our cells usually release a signaling protein called interferon. Think of interferon as a neighborhood watch system.
Okay.
If a cell gets invaded, it pumps out interferon to warn all the surrounding cells to raise their shields and stop replicating.
But Ebola bypasses that.
Ebola is ruthless. It produces specialized proteins that actively block the cell from producing or responding to interferon. It essentially cuts the neighborhood phone lines before the alarm can even go out.
Wow. And while the alarms are down, it starts targeting dendritic cells, macrophages, and liver cells. The Lancet paper actually mentions it causes islands of necrosis in the liver, basically creating dead zones of tissue.
Yes.
That sounds incredibly destructive.
Yeah.
But here's the big revelation that totally shocked me. We always associate Ebola with catastrophic blowing, right? The old name was literally Ebola hemorrhagic fever.
Right.
But the data from West Africa shows that hemorrhage, like actual bleeding, is surprisingly uncommon.
It's probably one of the biggest misconceptions out there. Bleeding occurred in less than half of the patients during the West African outbreak. The medical community has heavily shifted away from calling it a hemorrhagic fever.
Interesting.
Yeah, we now prefer the term Ebola virus disease, or EVD, specifically to downplay bleeding as the defining feature.
Oh, wait. I can understand some bleeding from the gums or, where an IV was put in. But if they aren't bleeding to death like in the movies, what is the actual mechanism killing the patient?
It is profound catastrophic gastrointestinal dysfunction.
Really?
Yeah. A few days into the illness, the virus triggers severe nausea, vomiting, and diarrhea, and the volume is just staggering. A single patient can lose 5 to 10 liters of fluid a day.
10 liters. That's over 2.5 gallons of fluid a day. The human body doesn't even hold that much blood to begin with.
Which is exactly why the result is severe hypovolemia. a massive critical drop in blood volume. The virus is simultaneously triggering an intense systemic inflammatory response.
Cytokine storm.
Right. Your immune system realizes something is terribly wrong and just floods the body with inflammatory signals. This causes your blood vessels to become highly permeable. They literally start leaking fluid into the surrounding tissue.
So it's a dual attack. You're losing gallons of water through your GI tract, and whatever fluid is actually left in your veins is leaking out through the vessel walls.
Exactly. And that leads to tissue hypoperfusion.
Meaning there isn't enough fluid volume or blood pressure left to pump oxygen to your organs.
Without oxygen, your kidneys, your liver, your lungs, they just shut down. That cascade of multi-organ failure and severe hypovolemic shock is what ultimately causes death.
It's like the virus pulls a fire alarm to distract the immune system, and while everyone is looking the wrong way, it drains the building's entire water supply.
That's a grim but very accurate way to look at it.
That is brutal. And if you think about the logistics of treating that, a hospital has to safely manage and replace up to 10 liters of highly infectious bodily fluids per patient every single day. The resource drain must be astronomical.
What is the supportive care required to keep a single patient hydrated and their electrolytes balanced during that acute phase is an immense undertaking, especially in under-resourced medical settings.
Right. Well, let's assume a patient manages to survive that acute shock. They get the IV fluids, their immune system finally kicks in, and they pull through. You would logically think the war is over.
You would think so, yes.
But the sources detail another phase that completely upends that idea. Here's where it gets really interesting.
Right, the concept of viral persistence. This fundamentally changed how we view recovery from Ebola.
Yeah.
We used to think that if a survivor's blood tests, specifically RT-PCR tests that look for viral genetic material, came back negative and they had generated antibodies, they were clear.
But the virus is still in the body.
It retreats to what we call immunologically privileged sites.
Wait, why do we even have those? Why would the body have places where the immune system isn't allowed to patrol?
It's an evolutionary trade-off. Some tissues are so delicate that a standard inflammatory immune response would destroy them. You don't want massive swelling and immune crossfire inside your eyeball or inside your central nervous system every time you get a systemic infection. So the body naturally mutes the immune response in those specific areas to protect them. The eyes, the central nervous system, and the testes.
Wow, those are like biological panic rooms. If the immune system is the police force, these privileged sites are like foreign embassies where the cops have no jurisdiction. The virus just slips inside and locks the door.
That's a great analogy. And because it's hiding there, survivors often develop post-Ebola syndrome.
Which is what, exactly?
Well, this isn't just fatigue. It includes severe joint pain, encephalitis, and serious ocular problems like uveitis, which is a really painful inflammation inside the eye that can lead to blindness.
Oh, wow.
And the persistence in the testes is incredibly concerning from a public health standpoint.
Yeah, the sources note the viral RNA can remain viable in seminal fluid, showing about a 4-fold reduction in concentration each month. And it has actually been sexually transmitted over 500 days after the survivor's initial disease onset.
Yes.
Over a year and a half later, a seemingly healthy survivor can spark an entirely new chain of transmission. And it's also been found in breast milk, right? Leading to transmission to infants long after the mother is supposedly cured.
Exactly. So if we connect this to the bigger picture, it means an outbreak doesn't end when the last acute patient leaves the treatment center.
Right, you're not done.
Not at all. You need an entirely new phase of post-outbreak surveillance. You need long-term survivor care, semen testing programs, and counseling to prevent these rare but very real resurgence events.
Okay, so what does this all mean for our ability to fight back? Because we are faced with a virus that cuts the cellular alarm wires, violently drains the body's fluids, and then hides in biological panic rooms for a year and a half. I want to look at the arsenal and the armor the medical community is building, because the NEJM article gets really deep into the recent clinical trials.
Yeah, the history of treating Ebola has been a series of immense efforts, followed by really disappointing results. During the West African outbreak, out of sheer desperation, several experimental treatments were rushed into trials.
Like what?
They tried convalescent plasma, taking blood from survivors, hoping their antibodies would help the sick. They tested small molecule antiviral drugs like favipiravir. None of those early interventions showed statistically significant survival benefits when tested in randomized trials.
But then we get to the outbreak in the Democratic Republic of Congo around 2018. They launched the PALM trial. And the way they did this seems ethically and logistically complex. The sources mention something called the MURI framework.
Right. URI stands for Monitored Emergency Use of Unregistered and Investigational Interventions.
A mouthful.
It is, but it's a crucial, specific ethical framework developed by the WHO. The dilemma is this. How do you ethically conduct a randomized, controlled trial with unproven drugs during a lethal epidemic, where giving someone a placebo or a lesser treatment could literally mean a death sentence?
Yeah, how do you navigate that?
While the MUI framework allows researchers to rigorously test these interventions while ensuring patients still receive the absolute highest possible standard of supportive care.
And the Palm trial tested 4 different therapies, right?
Hmm.
2 were older antiviral drugs, Zmapp and remdesivir, but the other 2 were monoclonal antibody cocktails,
Mhm.
MAB114 and RayGNA-EB3.
Yes. And the results were so decisive that the independent monitoring board actually stopped the trial early.
Wait, really?
Yeah. The patients receiving the monoclonal antibodies, 114 or REGN-EB3, showed vastly improved survival rates compared to the other 2 drugs.
So, how exactly do these monoclonal antibodies work? We know the virus uses that glycoprotein spike to unlock the cell.
Right, so think of the virus's glycoprotein as a highly specific key trying to enter a lock on the surface of our cells. These monoclonal antibodies are essentially like pouring targeted superglue directly into the keyhole.
Oh, that's clever.
Yeah, the virus is still floating around, but its primary weapon, its ability to bind and enter the cell, is completely neutralized. The immune system can then sweep in and just clear it out.
That is an incredible breakthrough for treatment. So, we basically have biological targeted drone strikes, but we also have a massive breakthrough in prevention armor.
Absolutely. The development of the Ebola vaccine is truly one of the great triumphs of modern virology. The standout is a vaccine called RVSV-ZIBOVB-GP.
Commercially known as ER-Vibo.
Right.
The sources mention it's a live attenuated vector vaccine. Can you kind of translate that?
Sure. It means scientists took a different harmless livestock virus, the vesicular stomatitis virus, or VSV, and genetically modified it. They removed its surface protein and replaced it with a glycoprotein spike from the Zaire Ebola virus.
Okay.
So when it's injected into a human, it can't cause Ebola, but it looks exactly like Ebola to the immune system. The body learns to recognize that specific spike and builds an army of antibodies against it.
And the numbers are staggering. In a trial in Guinea and later deployed in the DRC, the efficacy was estimated at 97.5% for people who developed the disease more than 10 days after vaccination.
Yeah, that 10-day window is critical because the immune system needs time to study that harmless spike and build the antibodies.
Right.
But once it does, the protection is profound. And they deploy this using a strategy called ring vaccination.
What does that look like on the ground?
Imagine a pebble dropped in a pond.
Yeah.
The pebble is the confirmed patient. Public health workers immediately identify and vaccinate every single person that patient came into contact with, family members, doctors. That's the first ring.
Got it.
Then they identify and vaccinate the contacts of those contacts. That's the second ring. You create a literal geographical wall of immunity around the virus, cutting off its pathway so it just starves and burns out.
That is brilliant.
They are also developing prime boost regimens like a 2-shot combination from Janssen and Bavarian Nordic to give long-lasting immunity. And that is crucial for healthcare workers who face constant exposure over years.
Okay, so science has essentially delivered the ultimate toolkit. We have the superglue antibodies to treat the sick, and we have a 97% effective shield to protect the healthy. You'd think the story ends there with a victory lap.
You'd think so.
But if you look closely at the DRC outbreak details, the armor has a glaring, massive weak spot, and that is human psychology and behavior.
Yep, this is perhaps the most sobering takeaway. All the virology in the world means nothing if you can't actually administer the dose. Outbreak management relies entirely on human cooperation in incredibly hostile environments.
Right.
Let's start with the physical environment. Because the infectious dose of Ebola is so incredibly small, doctors operate under a zero-tolerance policy for personal protective equipment.
You mean the full sealed hazmat suits?
Right. But wearing a heavy, non-breathable suit in the extreme heat and humidity of Central Africa causes rapid, severe heat stress.
Wow, so that severely limits the capacity to provide that intense fluid replacement care we talked about earlier.
Exactly. It's a huge logistical bottleneck.
And beyond the physical limitations, the societal hurdles described in the DRC outbreak are just tragic. We are talking about a region dealing with decades of violent armed conflict and deeply rooted mistrust of the central government and foreign interventions.
Imagine being in a community that has felt abandoned or actively harmed by authorities for generations. And suddenly, foreign medical workers arrive in terrifying spacesuits, setting up isolation camps. Experimental treatments and vaccines are being introduced.
Yeah, it's a recipe for panic.
Exactly. In that context of fear, rumors spread like wildfire. There were genuine beliefs that the treatment centers were places where people went to be killed, or that the virus was a bioweapon deployed for political reasons.
And that mistrust has violent consequences. The sources talk about actual armed attacks on Ebola treatment centers by local militia groups.
Yes, and even without violence, if a community doesn't trust the medical responders, they will simply hide their sick.
Right, which just fuels the.
Back.
But with Ebola, a corpse is at its absolute peak of infectivity, right?
Exactly.
Yeah.
So negotiating safe, dignified burials requires an immense amount of cultural sensitivity and trust. If you just dictate orders to a grieving community, they will reject you.
It's staggering to realize that a tiny, non thinking thread of RNA can exploit sociopolitical mistrust just as effectively as it exploits a host cell. It finds the biological cracks, the Niemann-Pick C1 receptor, the immune panic rooms, and it perfectly exploits the societal cracks of fear and conflict.
It is a stark reminder that medicine doesn't happen in a sterile vacuum. Defeating an outbreak is just as much an anthropological and sociological challenge as it is a virological one.
To summarize this incredible journey we've been on today, we started with the confusing family tree of the filoviruses, tracing their deep evolutionary history and the ongoing mystery of how they jump from bats to humans. We went inside the body to see how the virus tricks the cell into swallowing it, cuts the interferon alarm wires, and then triggers a massive systemic fluid loss busting, that Hollywood myth of people bleeding to death from their eyes. We explored the virus's terrifying ability to hide in biological panic rooms for over a year. And finally, we looked at the incredible scientific triumphs of monoclonal superglue and ring vaccination, which are ultimately held in check by the fragile complexities of human trust.
It's a profound story of human vulnerability, but also of incredible scientific resilience.
It really is. But before we sign off, I want to leave you with a final chilling ripple effect mentioned in the sources to ponder on your own. We talked earlier about the astronomical resources required to treat just one patient, the daily liters of fluid, the constant rotation of PPE, the specialized isolation wards.
It's massive.
Beyond the direct visceral horror of the virus itself, Ebola outbreaks completely overwhelm and cripple public health infrastructures that are often already stretched thin. The vast resources, the funding, and the medical personnel diverted to fight Ebola mean that routine health programs are simply abandoned. Clinics close. Vaccination campaigns for kids stop.
Yeah, everything else just halts.
So consider this. What if the deadliest feature of an Ebola outbreak isn't the virus itself, but the invisible shadow it casts? How many countless lives are lost quietly to malaria, tuberculosis, HIV, and childbirth complications simply because the hospital lights had to be turned off for everyone else? It turns out the real monster doesn't need to make you bleed to be devastating. It just needs to force us to look away from all the other threats waiting in the dark.